So can you see? Can you see the quiz? Yes? No?
Yes.
Get on your screen? Okay. So let's go through the questions one by one. First one, which of the following secretions are not produced as zymogens? Well, the only one that is protease, and that is so the pancreas can protect itself from being digested by its own enzyme. And so all the other items listed there, the lipase, amylase, and bile, are not produced as zymogens. So those should all have been selected. So why did I ask this question? Because zymogens are, the concept of a zymogen is really important. And I asked the question in sort of a reverse way. Instead of asking you what is a zymogen, I ask you what is not a zymogen, which to me is a more interesting way to put it. And either way, the answer should be clear. Okay, number two, amino acids methionine and cysteine are sulfur amino acids. I accepted any number of answers to that: disulfide bridge, disulfide bond, or covalent bond. And again, why did I ask this question? It's because we went over the table of amino acids several times, and every time I pointed out that these two sulfur-containing amino acids are particularly important in making bonds for the tertiary structure of proteins. And so that's why I ask you to tell me the significance of that. Anybody that has a question as we go along, please speak up. Number three, match the RNA type. Why is this question? Well, RNA is fundamental to understanding biology, and you need to know the different types and what they do. So mRNA codes for a protein sequence. Ribosomal RNA is a machine. It's a little structure that builds proteins. And tRNA, transfer RNA, specifies the amino acid that the mRNA is calling for. Number four, why did I ask about the pancreas? Because we spent a long time talking about digestion and how we digest our food, and the fact that the pancreas plays a major role in digestion, and it produces two things: enzymes and hormones. So that is what I asked, because we talked about exocrine function and endocrine function, and that one is to produce enzymes and the other to produce hormones that control the digestion, especially the transfer of digested food energy to the blood. And again, on question 5, I came back to zymogens. Is it true or false? Zymogens are inactive forms of enzymes, not necessarily glycosylated forms. So that is false. Number 6, we talked a lot. It's essential that you know about 5' and 3' ends of molecules, because they are referred to a lot as we go through molecular biology. So the way I framed this question was to say at the 5' end, what do you see? It's a phosphate group, and at the 3' end, you see a free hydroxyl group. again, it's really important that you know about 5' and 3' ends in order to understand the language. And then number 7 is about DNA replication. I showed you a video of this. We went over it. Beyond the video, and I emphasized the importance of knowing what each of these enzymes does. So the helicase unwinds the double helix. A single-stranded binding protein keeps, after the strands are unwound, keeps them from reannealing, from going back together. A primase attaches a short primer, and that primer is made of RNA. And it's needed in order for the next step, DNA polymerase 3, to do its job, which is to synthesize a complementary strand. And it starts at the point of the primer that was laid down by the primase. RNase H removes the primers when it's time. And DNA ligase, once the primers are removed, stitches the DNA together, joins the Okazaki fragments. So it's, again, I want to emphasize it's important that you know what the role of each of these enzymes is in DNA replication, because it's important that you know how DNA replication happens. Number 8, true or false, has to do with the structures of proteins. I asked this question about quaternary structure, and I assumed that if you knew the answer to this, you knew about the other types of structures of proteins. This was just one way of asking the question. And the answer is true. The quaternary structure is composed of 2 or more independent polypeptide chains held together by hydrogen bonds and disulfide bridges. And again, these disulfide bridges go back to the sulfur-containing amino acids. Number 9 was an essay. I wanted just a short statement about digestion of food. And what I was looking for here is that you recognize that what we eat is composed primarily of fats, proteins, and carbohydrates. The proteins and carbohydrates are soluble, and the digestive enzymes have ready access to them, whereas fats are not water soluble. And that's the role of the gallbladder. So I wanted you to tell me that the gallbladder solubilizes fats so that they can then be accessible to the lipases, which digest them, and they can be absorbed as energy. And the pancreatic enzymes, of course, act on all 3, fats, proteins, and sugars. And the last question, number 10, was again something that I emphasized. We went through an exercise in class indicating, and what I was looking for is that you know that C's specify G's in transcription, A's, T's, A's, and so on. And that, since I asked for the sequence of the messenger RNA, that in the message, there are no T's. Everything that would have been a T is now a U. So the sequence of the messenger RNA would contain 3 U's. Okay, any questions about this or the answers? Hopefully you have all checked to see if the grading of your quiz was correct. The speed grader often makes mistakes, and I try to catch those, try to correct them, but please let me know if I don't. The average grade for this quiz was 48%. The highest grade was 100%. The average time taken was 14 minutes. So that means that many of you took the full 15 minutes and therefore may have felt pressed for time, which makes me think that perhaps 20 minutes should be allowed for a quiz of this type. If any of you have thoughts about that, please let me know. But I am unhappy that the average for all the tests and quizzes so far have been about 50%, which means that there's a disconnect somehow between what I think I'm teaching and what you are learning. And I'd like to do something about that. Again, any suggestions that you might have, I would welcome. But let me just make a few suggestions right now, things that you might consider in studying the material in this course. First and foremost, you should focus on the slide sets and the recordings, because in them you will see that what I am emphasizing, and it will help you to focus on what I think is important. And what I think is important is the sort of thing I'll ask on tests. Secondly, attend Scott's workshops. That is probably the best way of understanding the material in terms of the big picture. Go to workshops with questions. Ask, in this particular set of information, what is the take-home message? What are we expected to learn here? And going through the materials, look at what I spend a lot of time at. So for example, I spent a lot of time on DNA replication. Okay, that tells you immediately that I'm going to ask questions about DNA replication. I spent a considerable amount of time on 3' and 5' ends of nucleic acid molecules. Again, that tells you I'll probably ask a question about that. You'll notice in some of the slides, I have something circled in red. Okay, if it's circled in red, that means I think it's important and you should know it. And the last thing I'd like to mention in terms of recommendations is that you spend time outside of class, outside of workshops, studying this material. The recommendation in the syllabus is that you spend at least 4 hours of concentrated study each week outside of class. So I think if you do that and you attend the workshops, ask questions, and try to be aware of what I'm emphasizing in the lectures, things should improve. So I hope that you can take this just as advice, and let's see going forward if the grades can improve. If the grades improve, that means you're learning more. Nobody said a word so far, so what are you thinking?
I have an opinion. I know that you give us 15 minutes to do the test, and also like I printed the slides and I make notes on the slides, but when I I'm stuck in a question and I'm looking through the slides to like find the notes that you've made and the ones that I made. Time passes so fast. So like I think time on the test is it's a fact of on these quizzes.
Yeah, I can tell that from this last quiz where the average time was 14 minutes. But you raise a good point, and that is that these are closed book tests. So you shouldn't be accessing your notes for taking the test. I guess I haven't been emphasizing that for each one. I mentioned that at the beginning. But I'll try to remember to do that from now on. When I make the test or quiz accessible, I will remind you that it is a closed book test.
Also, Mr.
Okay, I didn't know that.
But the other thing is, I think that perhaps there is too little time. The fact that the average time taken was 14 minutes out of 15. So I will try granting you more time, like 20 minutes for a 10.
Can you hear me, Mr. Yoder?
Thank you.
Okay.
Can you hear me? Can anybody hear me?
I can hear you very faintly. Could you speak louder?
Yeah, my mic is very weird today and for the past couple of days. But for the quiz, I have two things. The number 4, I got right, but it counted it wrong because I said secreting digestive enzymes and hormones into the blood.
Have you checked your quiz recently?
Yeah. Well, hold on. No, not the most recent. I got another notification, but I didn't know. I didn't check it yet.
Okay. Well, let me check that.
And then for the time, I think the time for me was good, but the reason it wasn't was because I didn't know. I struggled to remember a lot of the stuff. So it took me longer because I was constantly like going over what I put in and if I had the right thing and all of that.
Yeah. And that's why I think giving a little more time would be beneficial. Yeah. For example, for what you're saying, that you didn't have time to go back and rethink your answer before you submit it.
Yeah.
So I'll do that next time. And I think, yeah, the next quiz will be next Monday, and it will cover everything that I'm going over today, plus the last lecture, which was largely on mutations.
Okay.
And the CFTR protein. But the main point was mutations, and I'll talk more about mutations today. So I have a quiz similar to the past quizzes on Monday.
Okay.
And the reason for these quizzes is as spot checks, just to monitor how much you're learning from what I'm presenting. Anything else?
Yes, also for the quizzes, when the test times come, is there any way we can get like copies of the quizzes but like blank, because I can't go back and look at the quizzes once I check my grade?
Sure.
Because I think if I have the quizzes and can like quiz myself on all of the quizzes from that section, I think that would help me a lot.
Okay, let me just make a note to do that.
I second that.
Okay, that's 2 votes.
Same here.
It's a consensus.
It feels like the quizzes are extremely difficult, just in my opinion. And I feel like a lot of it has to do with the verbiage or the language or the way it's communicated versus the slides and then translated onto the test.
But
like
also, that's something Scott was talking to me about. Like, we like have to learn it in a way that we know different ways for it to be worded.
It just,
go ahead.
I was going to say, it feels as if the tests are not made up from our class lecture or from the slide. It has to do with just maybe the chapter that was read. They totally just don't align up language wise. So something that we may think is correct isn't correct because of the language that was used on the quiz is not matching the language that's used on our lectures or on any of the slides or on any of the notes given to us. So I feel like the percentage is failing a lot because it doesn't feel as if they are matching up. I think it's completely confusing. And I really like this class. I'm just bummed because I'm absolutely failing.
Okay, again,
I second that too.
making a note. The language used. It's an important point. It's something that I think about when I'm composing a test or a quiz. And what I try not to do is repeat exactly on a question what was presented in the lecture. And what you're saying is that throws you off, that you're looking to see something on the quiz exactly as you saw it in the lecture or on a slide.
Exactly. Yes.
Okay, well, we're going to have to compromise on that one because for me, if you really understand the material, you'll be able to tell me the answer regardless of how I phrase the question. But I understand what you're saying, that that's only partially true. And so I will keep that in mind and try not to deviate far from the way it's presented in lecture when I compose the question. So I'll try that next week and we'll see if it goes better.
Okay, I agree. I'm really enjoying the class. I just would
Me too.
like to be passing, and I'm afraid I'm definitely not going to pass if it keeps going this way. Yeah.
Well, yeah, I'm concerned about that too. I know that you're all trying, and I'm disturbed that the average grades are so low. So I want to do anything I can to improve the grades, but at the same time, improve learning. I want you to really learn this material because it's fundamental to understanding life, and you live life every day.
Maybe if you tried doing practice quizzes, that would also help a lot.
Okay, practice quizzes, you mean having more quizzes?
No, but
more.
like
No.
a study guide a study guide or like some sort of review worksheet where we can look through the main points of your presentations.
Oh yeah, kind of like okay, I get what you're saying.
Okay, I'm not sure I understand. The subject of study guides has come up before. I tried study guides last semester, and what I found was that students were focusing just on the questions that I posed in the study guide at the exclusion of everything else. And it didn't change the scores at all last semester. So I like the suggestion of sending you past quizzes before a test because I will, if I think something is important, I'll probably ask about that topic more than once.
And can you start adding page numbers or textbook numbers to what we need to learn for the quiz?
Okay, I will. As we go farther into the course, the text becomes less and less relevant.
Oh, okay.
Text page
Yeah,
number.
because I noticed in the beginning weeks we had like specific pages to read, but then now it's all based on lecture.
Yeah. And that's because it's hard for me to find exactly the material that I want covered in the text. I will look, and I'll make a note here to send page numbers for the various sections if there are such things.
Okay.
But as far as you spending your time studying, yeah, reading in the text is a way to consolidate the information, it's presented in a different way. But I would say that Scott's workshops are more relevant on most topics than the text is.
Yeah.
Go ahead.
I was just going to mention, I definitely think Scott's workshops are super helpful. It's just, I don't know. It's just putting out more time already for this class and then making it a double class with him. So it feels contradicting, and I wish possibly it could be like combined together at some point, or maybe explained a little bit thoroughly, or not maybe as thorough. I'm not sure, but it's as if it's taking on a whole nother class. I mean, that's how it is for me.
Yeah,
But
me too.
I'm just trying to keep up, honestly.
Yeah, and what Scott and I are trying to do is make your learning process as easy as possible. And we don't want you to feel like you're wasting your time. Scott, do you have a comment about this?
So no, I could certainly see how someone could consider a workshop to be like a second lecture. Because when you bring up a question to me, I do try and cover the topic in detail for you to at least close to the level that Dr. Yoder provides. Obviously, I'm no substitute for Dr. Yoder in experience and information, but I can provide you, and that's what I try and do, is provide you the level of detail that I think gives you a full comprehension. What I don't want to do is just, I don't know, I feel like it would be cheating you to just say, here's all you need to know on something, because I don't know what's on the exams. I don't know what's on the quizzes. And, you know, the more specific a question you come with, probably the more quickly, the more directly I can ask it, unless as I'm probing around, sometimes I see that there's missing basic information, and that may frustrate people because I will go back over the basic information, basic terminology, basic vocabulary, basic concepts, because if you don't understand them at the basic level, you're going to have a more complicated time at the advanced level. I will do what I can, and I will try and help people. I do want to see people succeed, and I want to put the information out in a digestible manner. And if it comes across too much as a lecture, I do apologize. I thought that that's kind of where people were looking for, but we can certainly adapt.
Yeah, I wasn't saying it was more like a lecture. It's more about the time. That's all. I'm down to sit a whole, I guess, another hour going over something that's more important or more descriptive or whatnot. But it's just, for me, I've already set time for this class. And so adding a study group afterwards is like, okay, well, I can't go to work then, or I can't go back to work. So it's just already cut out the time for this class. But I do think your workshops are 100%. They're freaking perfect. But it's hard to combine them both. And it is for me. And I don't know.
Okay, and I understand that, Erica, and I appreciate that feedback. I would like to point out that workshops, you show when you can and you leave when you have to. There is no, you have to be there 2 hours. I do 2 hours to provide people a window to pop in and out as they need to. Unfortunately, I can't sit here for, honestly, they won't let me sit here for, 5 or 6 hours a day. They only pay me for a certain amount. They only allow me to schedule a certain amount of time. So that's why I've tried to do 2-hour sessions 5 days a week to at least provide people an opportunity. And I realize it does not match up with everybody. I would also point out that there is that discussion board on the main Canvas page for your section. And if something comes up and you would like some kind of explanation on a question outside of workshop hours, I do look at those a couple times a day, and I will do my best to answer them for you. So I acknowledge the fact that we're adults, and a lot of people have jobs or families or pets or things that need taken care of outside of classroom and workshop. And I am, Dr. Yoder, myself, actually the entire STEM department, starting at the department head, are very concerned with trying to provide you opportunities. And that's one of the reasons we opened up this discussion board.
Yeah, I do think it's been extremely helpful. I don't know where the medium's gonna hit, because I know that I'm not the only one failing. And I know that we are all super trying our best. And it seems to just be something that's highlighted in class or over lecture. The quiz turns out to be completely different. But I don't want to keep beating a dead horse either. I'm just saying, like, I don't know what's going on, but the majority of us are failing. And we are sad about it.
I
I
think
feel like
that I had that same problem, like the exact same feeling that you're feeling, but I feel like maybe more class interaction, more questions during class. If you don't understand something, to ask right then and there. Maybe like listening to the lecture 2 or 3 times during the week. Like, it is really hard because I'm also failing, but like you have to figure out your schedule and what could possibly work to help you out, as well as the classes and the worksheets and the quizzes and stuff, because it's really hard. Like, it's hard for me too. I'm pretty sure everybody in here is struggling.
honestly, I think that that what's it called? That worksheets and like practice quizzes would help out a lot, because I cannot attend to the workshop
Yeah.
because of my schedule. And well, yeah, I believe basically that worksheets and like practice quizzes would help out a lot to like help us a lot with the exams and the quizzes.
Okay, that's a second vote for practice quizzes.
I feel like what Erica, like, this is what I got from what Erica was trying to say, is like not really to like take away the like amount of information and time that Scott spends, but more so like get that information in class rather than having to do it outside of class. So like be able to actually get that information during class rather than having to go outside of class to understand what we're supposed to be understanding in class.
I agree. I wholeheartedly agree and encourage that. So please, if I go too fast, or if you'd like me to go back to something that I went over too quickly, ask at the moment. I'd much rather have discussion in class than to have you go away not understanding, or to have to replicate that elsewhere. So please, I encourage you to speak up. It's crossed my mind that maybe I should go through slide sets twice. Go through once and then go through again, because with a little time to think about an issue, you might have a question about it.
Yeah.
Professor, I believe question 4 was not in the slides.
Question 4 about exocrine and endocrine functions.
Yes.
I think you're right. When I say slides, I was referring to illustrations in the lecture. Remember the documents that I sent you, and we went over the documents as though they were slides? The document about the pancreas, do you remember that? That's where that question came from. So if you were focusing on just slides and not the documents that we discussed in class, then yes, you would have missed that.
So just to reiterate that point, the very first paragraph of the cystic fibrosis, the pancreas document he posted, says at the very end of the first paragraph, the pancreas is a curious mix of exocrine secreting digestive enzymes and endocrine releasing hormones into the blood functions, which is pretty much directly where that question came from.
That's right. And the remainder of the period was an elaboration of those 2 points. We talked about specific enzymes and specific hormones and zymogens and so on. But the basic point was just what Scott said.
I think Scott's workshops are super helpful, and I hope he does not stop them. And maybe an idea, I don't think Scott would definitely give us answers to the quiz, but maybe if it's just an idea, just to send him maybe at least before a test, some information that we should super embed, even though you're giving all the information. But if you two were I don't know. It's up to you to send him the quizzes or not. I don't think I know that he would not say, okay, the answer to this would be number 5A or something like that. But the workshops, I do recommend. I don't know. I'm done for now, but it's an idea.
Okay. Yeah, so Scott, you and I should try to coordinate on this. Usually I don't make up a quiz until right before the quiz, and so I have no chance to send it to Scott. Once I do have it composed, I do send it to Scott, and he usually gives me a critique, and I modify the quiz after his critique, and then post it for you. But so we have a bit of a logistical problem here in that to try to give Scott advance notice of what's on a quiz before the workshop, like for right now, if you want to talk about what's going to be on next Monday's quiz, I don't know, and Scott doesn't know.
Right. As long as somehow, some way, I just think we're on different pages, and I'd like to somehow get us all on the same frequency.
Okay.
That's
Me
all.
Too. I would very much like to do that because clearly there's a disconnect between what I think I'm teaching and what you're learning. And if we can fix that, test scores will go up.
Agreed. Thank you, Scott, and thank you, teacher, for everything. It is a very interesting and enjoyable class. I know I'm not the only one struggling.
No, you're not. The average score is only 50% in the course so far. Now, there's always a few high scores. On this last quiz, somebody got 100%. There were a few in the 90s and 80%, but that's the minority. And I need to address, we all need to address what's going on with the rest of the class.
Professor, if we could also add like maybe 5 more minutes to the quizzes.
Yeah, I will do that.
Okay, and I got one last question.
Uh-huh.
Can we start from the beginning of the quiz to review when we get started?
Say that again.
Can we start at the beginning of the quiz when we start to go over it again? Because I missed the first 3 questions.
Are you asking for a second attempt?
No. Like right now we're going over the quiz, right?
Oh, after the fact. Yeah. Okay. So what?
Yeah, that's all I meant.
Oh, you want to go back to the first 3 right now? Okay.
Unless someone else has a question. I feel like someone else wanted to ask something.
I was just going to say, it was so helpful when you were going through your slides and you made summary slides at the end of your points, and you kind of put all of your important information on the end of the slide, kind of like here's a summary of everything I just talked about that you should know. Because I went back and I was able to read those, and it made a lot more sense for me to read the summary and make my own connections than to have to read every single slide and do that. So that was helpful.
Okay. Good point. And I will do that at the end of each section or topic. I will try to list what I think is the summary of that material. Yeah, thank you.
Another thing that I think would help is you grade the homework, because like last time I was doing it and I didn't know if I was right or wrong at the end of it, but then I did the quiz and I ended up getting one of the questions wrong. So I think graded homework would help.
Okay.
I know it's a big time ask, but yeah.
All right, I'll make a note of that.
All right, thank you.
Okay, this is a good discussion. I really appreciate your input. Anything else before we move on for today? All right, I'm going to take the quiz down and stop sharing. I want to move on to the next topic. You see how to cause mutations?
Yes.
You do see that? Okay. Okay. Now, last time we talked about mutations, specifically mutations in the CFTR gene. And I want to talk more about mutations now, but focus on a particular method for making mutations called CRISPR. Now, this slide shows, it's taken directly from the previous lecture. How do mutations occur? Well, there can be copying errors. There can be radiation damage. There are chemicals that damage DNA. And these are all bad if you don't want mutations. But what if you do? And that's where genome editing, or CRISPR, comes in. And you've probably already heard the term, the acronym CRISPR, because it's commonly in the popular media. And what causes mutations? There are a variety of, okay, so if you want mutations, how do you go about getting them? Well, in the past, you can wait for mistakes to be made in cell division, but you could wait several lifetimes to get the mutant that you want. So waiting for a natural occurrence is not an option. You can use mutagens like radiation, and I talked last time about UV radiation, how it causes thymine dimers. The dimers cause kinks in DNA, but again, these are random occurrences, and you may have to select or screen for a long time to find the mutant that you want. So use of mutagens is non-specific. Targeted mutagenesis, on the other hand, is just that. It's targeted. You get only the mutants that you want. So why is this so important? Because CRISPR permits any mutation in any gene in any organism. So to be specific, the procedure is site-specific, that is, it's targeted to just the gene that you want to mutate. And it's universal in that it works in any organism. Prokaryotes, eukaryotes, any organism can be the subject to this methodology. I should say that CRISPR, you see CRISPR spelled out at the bottom of the slide, clustered, regularly interspersed, short, palindromic repeats. That means nothing to most people. You'd have to know something about how they were discovered to understand why they're named with such a clumsy name. But I want to just give you a brief idea of how the process works and why it is so specific and why it is universal. I'm going back to this slide again to point out definitions here. What is CRISPR? It's bacterial DNA sequences that recognize specific RNA sequences. And the specific RNA sequences are what's important. Cas9 is an enzyme that cuts foreign DNA. And to use this process, that's all you need. You need the guide RNA, which you synthesize in the lab, specifically for the gene you want. And you add the enzyme Cas9, which makes a specific cut. And that's illustrated here on this slide. The Cas9 protein forms a complex with the guide RNA. The guide RNA is what you synthesize in the lab. So you have a gene that you want to mutate. You know the sequence of that gene because genome sequencing is very common these days. So you automatically, you just look in a database, find the sequence of the gene you're interested in. That might be CFTR, you look it up. You get the sequence and you make an RNA that's complementary to that and mix it with this enzyme, Cas9. And what you see then is that the guide RNA, in purple here, anneals to or attaches to the target. And when it does that, this little yellow sequence here, which is called PAM, tells Cas9 to cut the DNA. It makes a double strand break. And then you can have any number of things happen. You can leave it like that and you have a deletion, or you can put something in the gap. Now I have here two other illustrations of the process. We don't have to go over them in detail, but I have some definitions here on the left. gDNA or guide RNA, it's complementary to the target. The target is the gene that you want to mutate. It's synthesized in the lab. It's an easy process if you have the equipment to make a guide RNA. And the guide RNA then finds its complement, which is the gene you want to mutate. And in combination with Cas9, Cas9 comes along and cleaves the DNA. DSB is a double strand break. Now, I put a red circle, and I said that you should pay attention to things that I put a red circle around at the bottom here. And the reason is that it just makes the point that once you've cut the DNA, you can make an insertion, you can make a deletion, you can make a frame shift. If you make an insertion, it can be anything. It can be a gene from another organism. It can be a mutation, a mutated gene of the one that you've targeted. But you just have lots of options once you've allowed Cas9 to cut the DNA. So this is a bit of a summary. Let me just point out that, or to say once again, why CRISPR is so important. It has extreme specificity. In other words, it targets just your gene. There are few, if any, if it's done properly and the guide RNA is made properly, there are very few off-target events. And it works in any organism. This has been demonstrated from bacteria to archaea, viruses, eukaryotes, all kinds of mammals, plants, fungi, fish, and so on. CRISPR has been tried and CRISPR works. It's been tried on humans, it works on humans. And that leads us to ethical problems because gene editing in humans is considered by many people to be unethical. And in fact, some experiments have been done which have been declared by the scientific community to be unethical.
I have a question.
Yeah.
Where does CRISPR come from?
Bacteria. So that's a good question. CRISPR is part of a bacterial immune system. So bacteria have viruses, and the CRISPR sequences are designed to, if a virus attacks a bacterium, the CRISPR sequences remember the sequence of the virus. And they use this enzyme, Cas9, to cut it up, destroy the virus. So what humans have done is take what bacteria developed over evolutionary time to protect themselves from viruses, and they've used components of it as a gene editing system. And the two components, one is a guide RNA, and the other is Cas9, the enzyme that cuts DNA, but only cuts it where the guide RNA tells it to. That's the beauty of the system.
So CRISPR is just already in our bodies?
Can you say it louder?
So CRISPR is formed in our bodies when a virus is present?
CRISPR is never formed in human bodies. It's only ever formed in bacteria. But what humans have done is to take components of this bacterial immune system and use it for their purposes. And their purpose is to make mutations.
Okay.
And again, the beauty of CRISPR is that it only makes the mutation that you want. All other ways of making mutations are random. And so you make a whole lot of mutations in the genome, and then you have to sort through all those mutations to find the one you want. In the case of CRISPR, you know what you want, and you target it.
Okay.
Just some example of potential applications for CRISPR, and CRISPR is being used for all these things right now. Destroy the genomes of viruses. So for example, you could think of coronavirus, which we're going to talk about next. You can correct genetic flaws. So for example, in cystic fibrosis, it's already been done. You know what the mutation is in your CFTR gene. You can put in the wild type sequence using CRISPR technology. For crop improvement, stress resistance can be used to, in principle at least, rescue endangered species or eradicate invasive species, and reverse pesticide and herbicide resistance.
Wait, so say that last one, reverse pesticide and herbicide resistance?
Yeah, pesticides and herbicides are used widely in agriculture.
Yeah.
And the pests or the weeds can become resistant. And they become resistant because they undergo mutations. Well, you could reverse the resistance by reversing the mutation or putting back the wild type sequence. Okay, I don't have to go over that. All right, I'm going to stop sharing this screen and go to the next topic. Unless are there any other questions about CRISPR and why it is so important? It's just not me saying this. It really is truly revolutionizing biology.
So just to make sure I'm getting it right, humans can take...
I can't hear you.
Can you hear me now?
No.
How about now?
Yeah.
All right. So humans can take samples of CRISPR to make their own mutations to target a specific thing in any organism.
That's right.
Okay.
By specific thing, it's a specific gene.
A specific gene. Okay. I'm gonna have to study that.
If we go back to, can you still see the slide?
Yeah.
You see the double helix, the DNA. You see the forceps. It's plucked section of the DNA out. So that illustrates what CRISPR does. It cuts out a piece of DNA. Then you, the human, have the option of replacing that piece of DNA with something else.
Right, and that's how the reverse pesticide works, because it stops them from being able to be resistant to it.
Right.
Okay.
Yeah, okay, you got it. All right, I'm gonna stop sharing this screen. We've got about 15 minutes. Now do you see coronavirus?
Yeah.
Yes.
Okay. Does it help if I use the slideshow? You like that better than not?
Yeah, the slideshow,
Okay,
because I can see all the words better.
okay.
I haven't seen this.
Okay, you see that? Coronavirus, COVID-19, which you hear in the news all the time, is the name of the disease. The name of the virus is SARS-CoV-2. So we're in the midst of a worldwide pandemic, and I would like you to know something about why. How does this virus infect us humans, and why is it causing a pandemic when none of its relatives do? So on the left here, you see a scanning electron micrograph with artificial color showing the surface of a coronavirus particle. And the red things are proteins sticking out on the surface of the particle that look to somebody like a crown, and so that's where the name coronavirus, corona meaning crown, corona comes from. In the center, at the top, the blue object is a cross-section of a virus particle. And what you see, the red is RNA. You remember that viruses can have either an RNA genome or a DNA genome, whereas all forms of life, viruses aren't living, all forms of life have only DNA. But viruses have a choice. So coronavirus is RNA, and on its surface, the little blue things, little blue clusters, are spike proteins. Then on lung cells and many other cells in human bodies, there is a receptor that specifically recognizes spike protein on the virus surface. That receptor is called ACE2, which stands for angiotensin converting enzyme 2. And on the right here is simply a 3D picture of the tertiary structure of spike protein. You can see in it alpha helices and beta pleated sheets, which we talked about earlier. Now at the bottom of this slide, the infection process, the spike protein attaches to ACE2, which is an enzyme on the host cell membrane, and that starts a process called endocytosis, which we'll go into detail at some other time. By the way, you see these little speaker symbols on some of the slides? That's because I've taken it from a previous lecture where I had audio input. So if you click on that, you can hear what I said previously when I presented this.
Oh, excuse me.
Okay, this is endocytosis. Here on the left, the stars are the spike protein, the green are the ACE receptors. And when the binding occurs, it starts this process of invagination, which when complete, gets the virus inside the cell into a vesicle where it is protected. So endocytosis.
That sentence one more time.
Say that again.
Can you say that exact sentence one more time?
Yes, the attachment of the virus to the receptor starts a process whereby the plasma membrane invaginates, and it continues to invaginate until it can pinch off into a vesicle. It's called a coated vesicle, such that the virus is now protected. And it can then proceed to have the host cell replicated. So endocytosis is a mechanism that the cell uses to take in other large molecules or cell parts or even whole cells. And the virus hijacks this mechanism. It requires energy, requires that the cell spend energy to do it. And so the steps are first attachment, then membrane invagination, membrane closure, and finally virus-containing vesicle. The coronavirus gets into us. Okay, here. I want to talk a little bit about the receptor, angiotensin ACE2. What I'm coming to now is a rationale for why this disease occurs. Okay, angiotensin is an enzyme that regulates blood pressure. That's its function. It has no interest in serving as a receptor for a virus. And the diagram here shows angiotensinogen, which is the precursor in active form, is activated by renin to either angiotensin 1 or angiotensin 2, and these are called vasoconstrictors. They make the blood vessels smaller. And if that's if it's something that you don't need, it results in hypertension, high blood pressure. So the enzyme ACE, ACE, causes high blood pressure. And you may have heard of high blood pressure medications that are called ACE inhibitors. And what they do is just inhibit that process. What ACE2 does is convert angiotensin 1 or angiotensin 2 into another form, which is a vasodilator. That is, it makes the blood vessels larger, so blood flows more easily, and it mediates against high blood pressure. So in yellow type here, ACE2 converts angiotensin from forms 1 and 2, which are bad if you have high blood pressure, to forms 1,9 and 1,7, which are good if you have high blood pressure. Okay, I'm making a point about blood pressure here to let you know that ACE2, its only goal, its only reason for existence is to regulate blood pressure. It has no interest in viruses.
Wait, it's only to regulate blood pressure?
Say it again.
It's only to regulate blood pressure?
That's its purpose. That's what it was evolved for, to regulate blood pressure. And somehow it got mixed up with coronavirus. So I've called this, as fate would have it, strangers in the night. ACE2 is an enzyme. Its only purpose is to regulate blood pressure. SARS-CoV-2 is a virus. It's one of many thousands of viruses, many of which can attack us, we humans or other animals. SARS-CoV-2 has a surface protein called spike. By sheer coincidence, just happens to have an amino acid sequence that binds to the ACE2 amino acid sequence. So the point I'm making here is that this is an accident. The COVID-19 is an accident. It just so happened that this one protein that this virus has binds to ACE2 on human cells. So that's point one that I want you to remember, that there's no intent here. There's no desire on the part of human cells to get sick, and there's no evil intent on the part of the virus to make humans sick. It was an accident. It just happened. So why is SARS-CoV-2 so highly infectious to humans, whereas it has a lot of relatives that cause flu-like symptoms, but they don't cause pandemics? There's a lot that we don't know about why SARS-2 is so highly infectious, but we do know a few things listed here. ACE2 with the receptor binding is 10-fold greater to SARS-CoV-2 than it is to other SARS-type viruses. So what does that mean? It means that it takes far fewer particles to start an infection. So if it would normally take 100 virus particles to start an infection with normal binding, with 10-fold greater binding, it would only take 10. So the amount of inoculum that a person would have to breathe in to start an infection is much lower with SARS-CoV-2 than it is with other viruses. So that's one reason it's so infectious. Another reason is that it's readily, the spike protein is readily activated by an enzyme, again, from the human host cell, an enzyme called furin. An endoprotease cleaves preproteins into active forms. Furin is much more active on SARS-CoV-2 than it is with other viruses. And finally, this virus has the peculiar characteristic that it has about a 2-week latent period before symptoms appear. And that means that someone can be infected and carry a heavy virus load and express no symptoms whatsoever. And in the meantime, for 2 weeks, can be spreading the virus around to other people who then pick it up and don't know they have it for 2 more weeks. So that's one issue. Another is that there are symptomless carriers which can have a very heavy virus load and never express any symptoms at all.
So does ACE2 make SARS-CoV-2 have a stronger bond?
Excuse me?
Does ACE2 make SARS-CoV-2 have a stronger bond?
No, that's just a coincidence. That there are 4 amino acids in ACE2 that bind very tightly to the same 4 amino acids in the spike protein. And that's a coincidence, just happened to be that way. But this binding, because it is so tight, it allows for the virus to spread more efficiently because there are so few particles needed to start an infection.
Okay. So it's just a coincidence, right?
Just a coincidence, yeah. That's my main point here. Aside from this last slide, which tries to give a partial explanation for why it's so infectious, the disease itself just happened. Just a coincidence that spike protein and ACE2 have this tight binding.
Okay.
All right, I'm going to stop sharing that. Okay, I show you this. We're out of time for this period, so I can't really go into it, but I'm just going to introduce the next topic, which is membranes and transport. So we're going to talk about what membranes are, what the nature of a membrane is. It's kind of a mysterious structure. And we're going to talk about the function of membranes. How do membranes work to keep cells alive? So, as I said, I'll start here next time. And in the meantime, I will try to take your advice in preparing the next quiz. That will be on Monday. So Monday's class period will be only 60 minutes, 55 minutes, because I'll give you 20 minutes to do the quiz. Okay, any questions or comments about anything at all before we leave?
I just did answer somebody's question about the ACE2 and SARS-CoV. So I thought I would bring that up before, so they had a chance to look at it. It was actually Rachel that asked about it. So I've answered you, Rachel. You can take a quick look before the lecture gets shut down and the chat goes away.
Okay, I think I see. You start out the 10x affinity. Means that instead of 1,000 particles, you only need 100. Yeah, that's exactly right.
Yes, sir. And her question was directly before that. She asked if it affected it entering the body. And no, it would have to get into the body. I missed that answer, but it would have to actually get into the body before the ACE2 could work with it. So that part isn't necessarily correlation between the two, but probably there's a stronger bond between the two things because of the increased affinity, and it would be a coincidence. The natural world is full of weird little coincidences and happenstances where something is similar enough to something else that, and you'll learn this later on in the course when you talk more about enzymes, but and how they work. But it is possible for an enzyme which is only intended to work on one particular substrate to find another substrate that is quite similar to it, and it turns out it can have some effect on that as well.
Yeah. Part of the coincidence here is that there's a 4 amino acid sequence in SARS-CoV-2 in the spike protein. 4 amino acids in a row that bind very tightly to the same 4 amino acids in a row in ACE2. And it's those 4 amino acids that are responsible for this 10-fold greater binding with this disease than with most other diseases. And we're attacked by viruses all the time. Flu viruses are very common. Cold viruses are very common. But they don't cause pandemics. So it's just an unusual combination about this particular virus and this particular protein that's causing this pandemic.
Okay, so let me just make sure I got this straight. So ACE2 helps SARS-CoV-2 bind, and it makes it like a stronger bind, and that's why it's a pandemic?
Yes, that's why that's part of the reason. It's not the whole reason by any means, but part of the reason that it's so severe, that it's a pandemic, is this tight binding between ACE2 and spike.
Okay, thank you.
So basically it's like the bond is just strong, like way stronger and harder to break apart and destroy.
Well, that's true, but the result of that is that it takes so few particles to start an infection.
Oh, okay. It's easy to start.
That's right. Yes.
Okay.
If someone breathes in a few particles from the air, it's very easy for SARS-CoV-2 to infect. Whereas another flu virus, if 10 particles were breathed in, it would fail. The binding isn't tight enough.
That's
And you
that's
said they have the same 4 what?
There are 4 amino acids in a row that occur in both the spike protein and an ACE2 protein. And those 4 amino acids are responsible for this very tight binding.
But this is a big reason that they have the social distancing and they want people to wear masks, because the more they can reduce the amount of virus particles floating around, or the amount of virus particles in a particular given volume of air, the less chance you have of becoming infected from someone else.
Yeah, that's right. And regardless of what you read in the newspapers or what the government might say, this virus is spread through the air. I mean, it's spread through touching things as well, but it does travel as aerosols in the air. So masks are important. Okay.
Professor, I'm going to go ahead and check out.
Yeah,
Get my workshop set up.
Starts at 2 o'clock. So those of you who can, please join that. And I will see you all on Monday. I'll have a quiz. Well, the quiz will be available all day, and we'll have a 55-minute class period.
What? Never mind.
And Rachel, that's a great question that you were able to get in. This would be the kind of thing that you could post on the discussion boards if it came up outside of class, or you had this question come up outside of workshop times. That's the whole idea of being able to use that board. It's a good question.
Okay. All right, I'm going to end the session, and hope you all have a good weekend.
Thank you. Bye.
Bye.